Roots and how they work is hydraulic lift. Hydraulic lift is really cool. It's been shown about 20 years ago in desert plants, and it's been shown again more recently in grapes. There are some roots that plunge deeply, suck water up, and not send water back up into the plant. They actually feed the root system. They leak water into the fine root system at the surface. That's the most active root system. Roots need water to pick up a lot of stuff. They need water to grow. They need water and oxygen for all sorts of things. And they're much more effective in the upper 2 feet of the soil than they are in lower parts of that soil. But all the water's down deep. So these roots are sucking water up and redistributing it into the surface. And that's Vitis berlandieri again. So some of the berlandieri stocks do that, others don't. But it's a unique trait and a very important trait. So we'll talk about those as we go along too. Okay, we've already talked about this. I'm gonna skip over parts of this. It's gonna be on Dave's website too. It's welcome to post it too. You can look back through some of the stuff. And I've already said these things already because I tend to get ahead of myself all the time. But that's all right. You're gonna hear everything as we go along. Oh, there's the other one. Species and rootstocks vary in the way they regenerate roots. This is a really, really important viticultural concept too. When we talk about deficit irrigation, everyone's deficit irrigating to control quality. You will soon be deficit irrigating, whether it rains or not, to meet your water demands and needs. Because after this year, that inexorable roll of the political wheel has gone forwards. There's never going to be, in our lifetimes, the water where there was 5 years ago ever again. Not because of drought, not only because of people, but now politics too. It's going to be limited. It's going to be restricted. People will no longer be able to suck water out of their property, on the ground, on their property without restriction. That's going to happen for sure. It's going to cause a minor revolution, but that's going to happen for sure. Because it's a public resource. And it's being abused radically in parts of the state. And that's going to change. So we all have to start thinking about how we're going to use water more carefully. And I think even in wetter areas, understanding these things and using rootstocks properly is a major portion. So if the rootstocks and the species regenerate these fine roots differently, you can get in trouble from a quality perspective too. So let's say you're growing 5C or 1114 in your vineyard, and you've heavily deficit irrigated, really shortened those plants down and you're not giving them very much water. And you read the report that there's going to be a hot spell in a week and a half, and you say, well, I better get those root systems going again so I can take up more water and plump up those leaves, maybe generate a little bit better canopy. And lo and behold, some rootstocks don't respond to that. Once you cut them back to a certain point, it takes 3 or 4 weeks to regenerate roots, not 10 to 14 days, which is more typical. So you can really be in trouble. And that's what happens when we use those stocks that aren't well adapted to that. They're really not very appropriate for deficit irrigation. 5C is in that category, 1114 is in that category, Pure Riparian is in that category. There are a few others too.
How are you defining deficit irrigation?
About 60 to 80% of evapotranspiration, more or less. And it varies around the state. And Larry Williams' work has shown that you can, even in the San Joaquin Valley, you can give a vine 20% less water and still produce the same amount of crop. So we know we generally over-irrigate them. We can cut that back by quite a bit. You drive around areas and look at vines at this time of year, and if they look yellow and sort of gray, that's not a good sign. Those have been overstressed. They shouldn't look like that. There should be some active photosynthesis going on. If you have too much growth, you should have planted your vines further apart. You shouldn't be cutting them back and abusing them. There's a certain level of pain that they can take and others they can't. It changes the phenol profile, the color profile, all sorts of things that you really don't want to affect that radically. So you can overdo it, and I think that's another sort of issue too. So we talked about the fine roots, some don't. So 110R produces fine roots, but very, very sparsely. 1103 is sort of an interesting plant, and we bookended here inside of 1114, which produces lots of fine fibrous roots, not many good structural roots. 1114 is on the worst side. 110 is probably on the best side in terms of larger structural roots, but fewer fine fibrous roots. And 1103 does everything. Lots of fine roots, lots of medium roots, lots of deep roots. It's in fact a bad rootstock for dry farming oftentimes because it sucks up all the water very quickly. So if you're truly dry farming and you had 110 and St. George and 110R side by side on the same soil with the same amount of rainfall, the plant with the best leaves at the end of that season would be 110R. It would survive longer with less water. So it partitions water differently again. And the 1103 and the St. George will be a whole lot better than 5C or 5BB or 3309C or all sorts of other stocks But they would have far more leaf drop and far more effective growth at that point if you left them the same way. So that's a root system that's just taking up water rapidly. Freedom would be in the same category. Own roots are in the same category. They have all these different well-branched and well-ramified root systems that take up water more quickly.
In the propagation of the plants, is it better then for the nurseries to be producing bare root plants because of the restructuring of the roots, or green grass?
There's a good question. That could be another large argument we could have here tonight, particularly if there are nurseries in the audience. The best possible plant is a good, strong, dormant bench graft or dormant root. And that plant should be trimmed, unless you're going to plant it carefully. So those of you who both dabble in viticulture and horticulture, and have planted roses or fruit trees or whatever else in your backyard, realize you dig a great big hole, you put a little mound in the center, you tease the roots down the side, try to spread them out, try to point them downwards. We don't do that with viticulture very often. But we can compensate by cutting all those roots off. So you can take all those roots that came with that dormant rooting or dormant plant and trim them back to ½ an inch or an inch. Inch is probably better, and plant them in the soil that way. So you don't have to worry so much about if they're bent. But if they're long, and you bend them back up like this in particular, or even like this, those plant systems are damaged because they don't generate more of those roots. The ones that they come with are the ones that they stay with. It's a very strange phenomenon. In fact, I have a student working on that right now. Why does that happen? Why don't more roots compensate and come out and solve this problem? Why does J-rooting kill these plants? And it does, over and over again in the state right now. It's a big problem. It's really, really associated with potted vines too. Dormant potted vines, the worst possible plant. So how do you solve the problem? Pull them out of the pot, trim the roots off, put them in the ground. Okay. If they're potted and not dormant, then you're in that sticky situation of having to tease the root system apart and really make sure it's not spiraling and not kinking the plant. Really, really important. For all the reasons we talked about it so far. Okay. The density in the profile differs too. So there's rootstocks that are all shallow, and they're all the roots in that top 2 feet. Rootstocks that are primarily deep, all the major roots are down 4 feet in the ground or further, like 110. And rootstocks that do both, that fill that whole profile. And they all behave differently. And this is always to show people that if you want to see really nice backhoe kits, without a backhoe, go to Chile, particularly in the desert region in the north. I gave the big talk there one day and told them all about the rootstocks and how the root systems are going this and that and the other, and they sat there believing me. But that night, they had people out digging up rootstocks to prove to me that it was either right or wrong, or at least expose my untruths, I guess, in some ways. And it's amazing how they dig these things. They dig them all the way down a row between 2 vines, across the row, back to the other side. You can see the entire rooting profile. It's really fantastic. Incredible.
You're right.
I got it right. And it's actually based on a lot of studies. Not a lot of replication, but a lot of studies that have actually dug up these vines. And here's 420A, a fairly shallow root system, floating up on the surface. And here's 110R. We've gone through and gridded these things and counted roots and measured their diameter in the soil. So we know how these work in the field. We need to know how they work otherwise. Okay. Now there's one more about grape roots. Root behavior and structure will have an impact on downwardly mobile insecticides. So if you believe your crop protectant advisors or not, one of the things that's coming out more recently is we can solve phloxera, we can solve nematodes, we can solve all sorts of things with the ample use of imidacloprid and other neonicotinoids, and we just dump them in the ground or on top of the plant, and they'll go down to that root system correctly. Actually, it depends on how the root system's working. Remember, it's not a very strong sink. It's not a very strong magnet for stuff. So stuff doesn't really go down from the foliage into that root system very effectively. And it's not taken up very evenly either through different root systems, depending on how you've treated them and whether those feeder roots are ready. So you have to remember all those things if you're going to apply these insecticides. How are you going to get them in the roots? Make sure that you're not out-competing the roots, number one. How are you going to keep them in the roots? Make sure the roots are active when you're applying these things. Make sure they've had a pre-irrigation. You're actually getting some feeder roots. And the Riparian-based stalks will spring out quickly from that irrigation, and the Berlanderi ones will probably be a little slower in terms of how you're going to use them. Okay. So this is sort of cool. This is 101.14 and 110R under drought or not. And it showed us a lot of things. And one of the things it showed that under dry conditions, in wet conditions, you have water in 110R going in and out at the same rate on these major roots, these large superized structural finger-sized, not little feeder roots, but larger roots in the soil. And when you put it under drought, it stops letting water go out of the roots as much. It holds it in. It changes the whole process. 114 doesn't care. Just keep dumping. In fact, it dumps more out under drought than it does under water, without water stress. So they behave very, very differently. It's really important we know these things, so then I can start saying when we're breeding, what do we select for, what are the most important characteristics, and how can we sort of define these stocks for different situations. Okay, so which rootstocks to use? This is probably the only slide you'll look at again in Dave's deal, and it's an important one because remember, it's impossible to make the best choice. There's no best choice for rootstock selection, ever. There's always lots of bad choices. And I think that's the most important. If you don't know what the bad choices are, you start accepting that at the nursery. And usually it's because you've ordered too late, and then they have lots and lots of 114, but nothing else. Or lots and lots of 1103 Paulson. 1103 Paulson's a very good rootstock, but it's a very bad rootstock for close spacing on deep fertile soils. And those two rootstocks are probably 80% of all the stocks used in California, and they're used on all types of soils and situations. Why? Because they're available. They both propagate beautifully. They're not necessarily the best stocks. That's the key thing. And in some cases, they're the worst choice. So dry farm situation out in a dry climate, anywhere in fact, where you're getting less than 25 or 30 inches of rain consistently, you should probably not be using 10114. 1103 Paulson. Let's say you decide to give it up down here and move up to the north coast, maybe into Mendocino, in the Anderson Valley, for instance, on a nice fertile area with lots of water. It may be not a very good stock if you're going to put those stocks too close together, or those roots, plants too close together. So it's a much more vigorous plant. So those are the key points there. So the Riparia-based stocks are what? 5C, 10114, 1616C, sort of 3309C, but it's a bit more rupestris-like, sort of in between. So those stocks, they're all shallower roots, much more water sensitive. They have lower vigor. They have earlier maturity. This is actually going to become important at some point. And one of the things we're selecting for now in our new varieties is hastening maturity. Why are you doing this for? So, because if you get the crop off earlier, you can save 2 or 3 or 4 irrigations per season. You could probably save a lot of water. And that's one way to approach it. Can you make the same quality? I don't know. The hope is yes, but it's one of the things we like to think about and select for. So that category of stocks definitely hastens maturity. Moves the vine forward, definitely, without question. The rupestris-based ones, broadly distributed root systems, relatively drought tolerant because of that huge root system, moderate to high vigor, mid-season maturity as they go through too. St. George, 1103 Paulson, AXR, we don't use anymore, but it behaves just the same way. Home roots behave just the same way, and 3309C. So those are the sort of ones that are intermediate, somewhere in between. Not really drought resistant, but maybe more drought adapted or drought tolerant because of their ability to suck up more water. The Berlandieri-based ones, 110R, 140 Ruggieri, we haven't used that much yet, 420A surprisingly, and 5BB to some extent too, all behave more like Berlandieri. So these are stocks that are deeper rooted. They're by nature more drought tolerant. They're behaving more like Berlandieri too. They have higher vigor that they induce oftentimes in the vine. That comes from the rupestris group too as well. And they delay maturity. They slow down that ripening process. And in fact, the phenology of that mother vine is interesting. So if you come to Davis in September 15th this year, September 20th if you like, wander up there to our collection and ring me up on the phone and say, where's that collection? I want to see this with my own eyes. You'll go out there and look, and you'll see the Riparia-based plants are beginning to defoliate by the first part of October. The vines are slowing down and dropping leaves. And if you come back again in 4 or 5 weeks, at the end of October, middle of November, you'll notice that the vines that are Berlandieri-like still have active leaves, they're growing like mad, and still going just fine. Same amount of water, same soil, side by side. Completely different. There's probably a 4-week, maybe even longer than 4-week difference between the maturity of those things and how they develop. So that's good to know. You can exploit that. You can think about, well, how does that fit my vineyard situation? What do I want in terms of a ripening cycle? And how should I be using these things more effectively? Okay, we've talked about this enough. The key thing here, I think, is how, if we can uncouple a drought resistance from excessive vigor. So maybe drought resistance is just excessive vigor. Then we can go back to Dave's example where Topless Creek said, my God, 1,500 vines per acre is way too many. I better go down to something more reasonable and stretch them apart so they can use that soil more appropriately. You can use that sort of both directions, think about how they're spaced. But it's important because we can produce much more vigorous rootstocks. I've been selecting stuff that have roots that look more like carrots than grapevines. They plunge straight down, they're much thicker, they branch out laterally, that might be excessive. And those are things like Dog Ridge and Ramsey, plants with very excessive vigor. And if you put them on close spacings, no one's going to be at all happy about that. It's going to be a different sort of utility. So you have to sort of think about both aspects and how they work. Okay. And they really are different. You can almost see this now, right? It's almost dark enough now. Here's Ramsey on the right and Repair Gloire on the left, side by side. Big, thick, large, plunging roots and very thin, fibrous feeder root systems. And we've been chasing after this idea that it must be the depth that is the most important thing, and now we're sort of rethinking it. It turns out that girth, size, and number of roots, so fewer but much larger roots is probably what you were looking at. And that's what we're focusing on now when we're selecting for these things. This graph you can't see very well either, but it has all the major rootstocks side by side, with and without water. So we grew them all in a field plot. The first season with water. The second season we stopped irrigating half of them, left the other part alone. And this only showed me that they behave the same under drought or dry conditions or wet conditions. So the root angles, the root depth was the same, which is good. That means it's genetic. It's not just environmental. In a dry year they go deeper, in a wet year they stay on the surface. No, it really is genetic. There's something we can use in this process. So we've been trying to use that, and we've been measuring roots like crazy and thinking about how we can genetically map this trait. So what does that mean? We need to find markers, DNA markers, that will be associated with those behaviors. And I thought it would be easy when we first started this. And the hard part is, how do you measure the roots? So I've probably had 4 or 5 students who have not been able to measure roots because they're too particular about how they measure, in fact. They're going through and measuring every single root. And if you look at any of these root systems, there's some deep roots, there's some shallow roots, there's some in between, but there's a preponderance of roots. And it goes back to the idea that Mendel was a fraud. We all think about Mendel and biology and things and think about how that data must have been real. That data was mostly fabricated, and it was fabricated because it was categorized. So there was variation between wrinkled peas and non-wrinkled peas. There was variation between pink ones and white ones. He disregarded that. He said, they're pink, they're white. Grade them in those categories. And that's what I told the students. There are 3 classes, flat, deep, and medium. And they said, no, there are more than 3 classes. You have to measure all these roots. I said, no, you can't. Your eye is doing that for you. And if you use your eye alone, it works. If you categorize them, the genetics of this work out. If you don't categorize them, if you measure them all, the genetics don't work out at all. There's no indication there's any genetic control when you measure all the roots. So it's been tricky to work with, and we've been trying to find it so that eventually we can grind up a seedling and tell whether it's going to be deeply rooted or shallow rooted and put it in the right categories. So I don't want to save all shallow ones or all deep ones, but I want to keep a mix of these things as we go through. Salt turns out to be really important too. So where do we have salty problems? Anywhere where it doesn't rain enough in the winter to leach salts down deep in the soil profile, which is almost all of California south of Lodi. So if you think about how we're going to manage salinity at the same time with drip irrigation over long periods of time, we're going to be in trouble. So we need to start thinking now about how to address that in the next generation of vineyard. So if you go through well, I guess you could cultivate extensively and make sure you didn't plant the exact same part of that drip zone when you replant that vineyard. But there's going to be salinity problems associated with the drip irrigation that we haven't really addressed yet. Through fertigation, through application of water, through water quality issues, and not enough water leaching that stuff through. So Kevin Fort's really been working hard on this problem and trying to understand these things, and we've been looking a lot at these southwestern species. And it turns out we now have plants that grow in 1/3 seawater. Pretty salty. They don't grow very much, but they don't die. And we have stuff, of course, that croaks very rapidly at that. So we've gone through and started to select these things, and we found 2 big hot beds of that behavior. One is in the Red River of Texas. The Red River really is red. It turns out it's mostly silver if there's no water in it. When it dries down during the drought, it's all salt deposits. It's amazing. And if you look through that spot, there's a species there. This is this Vitis duaniana and Vitis acerifolia that are sort of funny parts of this middle part of Texas material that are remarkably salt tolerant. And the other spot is right over the hill here, and a little bit further on back in the east. If you go to Pahrump, Nevada, some of you have probably had a very poor time at a restaurant there. There's only one, and it's not a good restaurant. It's a horrible place to stop in Pahrump, Nevada, but there's a big salt flat there. And it's amazing. It's called Ash Meadows. And if you go out there, there's Vitis gurdiana growing all over the place. And when you walk out to those plants, the soil crunches. Those plants are remarkably salt tolerant. And it turns out that they're not really sodium chloride there, it's gypsum. It's a gypsum flat. But it still has this amazing ability to tolerate extreme ionic pressure. Claire's work is also looking at all this material I've been collecting over the last 25 years to define what it is. So if you go over the border here, so if you collect grapes, you're driving along in your car, if there was a guide, there should be a guide to grapes, so I'm going to do that at some point. As you're driving east across the desert, you're going to read Vitis californica to the north, Vitis gerdiana down here, and all the way to the desert, Vitis gerdiana, except they say there's none out there, but there's plenty of it. And as soon as you cross the border into Arizona, it says Vitis arizonica. And it says Vitis arizonica all the way to Texas. And it turns out it's actually 5 different species. And they're all intermingled together. And it's really important we know which ones are which, because that way we best travel and track these different characteristics and traits. So that's coming along too. And we're getting pretty close. I'm going to show you these funny zones where Vitis arizonica persists and exists. It's actually a blend of gerdiana, berlandieri, rupestris that used to grow there, and another species coming up from Mexico. We're not quite sure which it is at this point. And remember that all those plants that were around this area in the past, forming some of these hybrids that have occurred, are now mostly gone because of urbanization and highway departments. It turns out the highway departments hate wild grapevines. In fact, they hate any perennial plant. There's a huge crisis going on for you plant lovers in the crowd. The highway department has decided that grasses are easier to manage along right-of-ways everywhere in the United States, and no broadleaf plants that exist. So they've been spraying all those areas with 2,4-D. When you collect wild grapes, almost every single plant has 2,4-D damage on it, some place or the other. And you'll notice there's fewer and fewer ones. It's a big issue, actually, because we need them as viticulturists. We need these plants. They're important, and they're being exterminated as we go. So it's interesting. There's the Vitis duaniana. It's in between this zone, between the candy cane grape, mustangensis, and this Erythrofolia grape on top, as you can see. Sort of a weird little zone there. That's also a funny spot. So I've been lusting after these grapes for about 20 years. I've been back to the same spot 4 different times. The last time I went, I called ahead and found out who owned that property because I was fed up with not getting over this very, very serious fence. In Texas, it's probably a bad idea to jump over that fence. Not so bad in Oklahoma. California, there's no problem whatsoever. But in Texas, it'd be not such a good idea. So it turns out it's owned by the Comanche Nation. And they were very nice, and they took me around the property. But in the intervening 5 years, 5 to 7 years that I hadn't been there, they'd stopped grazing, there weren't any fires, there was a major drought, and almost all those plants had died out. It's amazing. So the really important germplasm that I have a few bits and pieces of is now gone. Amazing. Okay, I've told you this already. I'm not going to tell you that. This is interesting. So on these graphs, this is Ramsey or Salt Creek. This is small potted plants being exposed to 25, 50, 75 millimolar chloride, sodium chloride. 75 millimolar is about 10% of seawater, to give you some sort of perspective there. Pretty salty. 25% isn't much. And we did this over time. So with Ramsey, we started increasing the concentrations, and it worked out well. Over 3 weeks of treatment, what we thought would happen should happen. And these charts actually show the amount of chloride get up into the leaves versus being partitioned in the roots or going to the stems. So these plants that are sodium resistant, or not sodium, chloride resistant, are partitioning sodium chloride that comes into the roots. They're putting it into the root system. They're preventing it from going up from the top. So as we can overcome that with more and more salt.
I'm sorry for interruption. What does a vine look like that has too much salt because of this drought?
It looks just like you cut the roots off. So it burns back from the edges, sort of interveinally, and you'll see brown tissue on the edges. It's not uncommon around here, and if you want to go over the hill, you'll see even more of it.
Come to my vineyard, I'm thinking that's what we have.
Yeah, very likely. It could be PD, and it could be fungal rot as well. So it could be a lot of different things.
But so it's browning around the edges. And what about the coloration of the leaf itself?
If it's a red variety, it'll get red before it turns brown. You'll see a little red halo around that brown area. With the white variety, it'll be more chlorotic.
Okay, thank you.
Yeah, ask questions if you have any, not a problem. If you go 6 weeks with Ramsey, there's no salt resistance. So over a much more stringent period, with more salt and more time, that resistance breaks down. And the reason I told you that is because we have species that doesn't break down. Right? So you can do both. You can have great salt resistance and have it persist over longer periods of time. This form of berlandieri does that. This form of gerdiana does the same thing. It's able to tolerate much more salt and work with it. And we've now found some stuff that actually does the one step better. It doesn't let the salt into the root system. So the partitioning idea means it's taking up salt and puts it in the root parenchyma. It doesn't get up in the foliage, it doesn't kill your plant. But eventually, it's going to take up so much salt that it's going to kill those roots. And they're going to start being damaged too. So we need to find one that does both at the same time, and we've started to find those. And here's pictures of the ones we did in 150 millimolar in the third seawater. They're alive. They're not very happy, but they're alive. Most of the plants were dead. So we took about 600 plants and put them in those situations, and a few species are actually tolerating that at the same time. So we have to have both. As a rootstock breeder, I have to have both good drought tolerance, a good drought resistance on top of that, to think about where the rooting angles are going, and we have to add in salt resistance. It has to be in that same background. If you're up in Mendocino, Sonoma, it's not going to matter. But if you're down south of Lodi, anywhere where there's less than 15 to 20 inches of rain per year, you're going to start being concerned about both those phenomena at the same time. If you dry farm, you may not have to worry about it. Right? If you're not applying water, you're not actually applying water to those plants, there may not be a need to worry about salt resistance. But if you're irrigating to bring your yields up to a place where you can actually survive and grow and control quality, dry farm vines are actually don't make very good quality fruit in really dry, hot years because there's really not enough canopy to protect that crop. You're going to start seeing very distinct vintage years if you're dry farming in a hot climate because of the way the vine will grow over time. So we need a little bit of that water. So I think those two things fitting hand to hand are going to be important. So we had forms of Acerfolia that survived that 150 millimolar, forms of Duaniana, forms of Gerdiana, this Scotty's Castle Gerdiana. Hope nobody's here from the Forest Service, or if you are, you're retired from the Forest Service. That's from the Death Valley National Monument there. Scotty's Castle. Just open the door and pull in the plants. Just drive by very quickly. We all know the grapes grow better if you prune them, right? Ahead to the back a little bit. No. Okay, that's enough of that. And I don't know when you want me to talk about Pierce's disease, but I'll sit down and wait. Well, I think Chris is going to talk too.
I think we were going to switch it for you
Okay.
So that you can make your dinner reservation.
Ah, okay, even better.
And we'll put you last, Chris. How's that?
Sure. Dave.
And we'll
No, please.
Andy, a question? In your presentation.